So, moving on to the second session, part of our practical sort of seminar, right? Practical number 6, identification of parts and functions of the light microscope and use of light microscope to observe specimens. Okay. Then all light microscopes have 3 basic components. What are the 3 basic components? All light microscopes have 3 basic components. Before that, take out your resource books. So this is the practical, right? Before that, in your resource books, you have something more from the microscope that you need to know. Page number 22, online students, resource book, page number 22, we have the contribution of microscope to the expansion of knowledge on cells and cellular organization, right? So we talked a lot about biological molecules, right? After biological molecules, what is the organization level? Organelles, right? Organelles, then cell, right? So we are going in the nice order. Without microscopes, are we able to study the cellular structures? No. Our naked eye has a limited capacity to see small things, right? You will see there's a value resolution power, there are some parameters, right? Our eye is not able to comprehend or see these small cellular structures, but the microscopes and their invention gave us the opportunity to sort of like the doorway to study these small structures, right? These small microscopic structures, right? So that's why before we start studying about the cellular small structures, we need to know how are we going to study those. That's with the help of microscopes, right? So we have to have a basic idea as biologists or in the field of biology. Microscopy is very, very important, right? So even in medical field, there are subjects called pathology, right? So the study of tissues is called what? Study of tissues? Histology, right? Histology is like a basic science, right? We apply the study of tissues into a subject called pathology, right? Vyadhi pathology. In that, for example, if you suspect someone has having a cancer, right? Breast cancer, we take like a small sample, right? Cell sample like using a needle, and we send that sample to the pathology lab. Pathology lab, they mount it on a slide and observe it under the microscope, the pathologist, and he'll say, "Methana cancer cells then at the beginning of a cancer okay, he or she will give a report, right? So that's one of the applied sciences of this histology, right? Microscopy, we are applying. And microbiologists, right? In microbiology, so let's say a patient comes with an infected wound, right? Kakule, sarava pus and an infected wound. What do we need to do? If we suspect it's bacterial, we need to give antibiotics, right? Ape hema nikam antibiotics then naha, right? There's a real problem in the world called antibiotic resistance. These bacteria are evolution, right? They have got some new, let's say, skills to not die from antibiotics. They have evolved, right? They are resistant to antibiotics, right? So අපි දැන් ඉස්සර වගේ හිතිමනාපෙට antibiotics දෙන්නෙත් නැහැ, prescribe කරන්නෙත් නැහැ. Even in the hospital we don't give, because it's a real problem. There will be in the future, there can be very strong bacterial species or fungi, protozoa can be also there, that won't be killed by any antibiotic. So that's going to be a problem. Antibiotic resistance, one of the biggest reasons is antibiotics prescribed karne, right? You can imagine the bacteria are getting like they are experiencing the same antibiotic again, right? Because people are using it. So with time they are going to be resistant to it, right? So as I was saying, so in the hospital, before we give an antibiotic to a bacteria in the blood, right? You can get fever, bacterial infections like sore throats and your sinusitis, pneumonia, right? Or even in a wound, we do something called antibiotic sensitivity testing, right? We take a sample from the wound and we send it to the lab. Then we first observe under the microscope, right? Then we do something called culture, right? We get like a big population of that bacteria. Then we are going to put some antibiotics to it, and there's going to be a report, right? You haven't seen, right? There's something called antibiotic sensitivity, right? Lab එකේ antibiotics නිකන් let's say 10 common antibiotics 10ක් දාලා බලනවා මේවා answer කරනවා. එතකොට එනවා resistant sensitive කියලා එකක් එනවා. එතකොට ward එකේ doctor he'll see the antibiotic sensitivity report and say අන්න මේ antibiotic එකට මෙයා sensitive. එතකොට මොකද කරන්නේ? එයා resistant ඒවා දෙන්න යන්නේ නැහැ. එයා sensitive එක විතරක් nicely අර නිකන් specialized weapon එකෙන්ම ගහනවා. right? නිකන් අර වටේ තියෙන නිකන් ගහලා බල ඉන්නේ මේක හරියයිද මේක හරියයිද කියලා. නැත්නම් precise එකෙන්ම ගහන්න ඕනේ. නැත්නම් මොකද වෙන්නේ? ඉස්සරහට තව resistant වෙලා මූ තව strong වෙනවා. So that is another application of this microscopy, right? To observe microorganisms and to identify කවුද මෙතන ඉන්නේ? malaria ඉන්නවද? right? What else? There are some other diseases that we can identify just from microscopes. Microscope එකෙන් බලලා. Right. So today we are mainly, microscopes, we can divide it into the type of, let's say, the wavelength, the type of wave that they are using. Based on that, we can classify microscopes as what? Based on what sort of waves they are using, right? We have to use some kind of a wave, no? So a wave goes and reflects and come back to whatever the sensor is, right? So if you are using visible light, right, those kind of microscopes using visible light are called light microscopes, right? So light from a source reflected on like something, then comes to your eye, then you can see. That's light microscopes. Then with technology, we have developed another type of microscope, electron. We use a beam of electrons that is going to be reflected on something and going to come to your eye. No, sorry, not to the eye. We can't see electrons. It's going to be detected by a special sensor. Okay, special sensor. Those are electron microscopes that are connected to a computer, right? Okay, image එක හදන්නේ computer. I'll show you some electron micrograph images as well. Okay, so come to the resource again. Read light microscope. Okay, light is passed through the specimen and then through glass lenses, right? There is a specimen that is what we are going to observe, that is going to be mounted here, right? Then there's a light that is going through the specimen and then some glass lenses, right? Glass lenses then hitting your eye, right? So lenses, what do? They refract. What do you mean by refraction? So I'm again physics, right? Lenses do what? We usually use what kind of lens? This sort of biconvex lenses, right? Lenses do what? They bend light, no? So if this is the specimen, this is a light ray, they magnify it like this, right? This is what? Refraction, right? This is refraction. So glass lenses are used and they do what? They bend light through refraction. The simplest microscope is a single lens. So this is a simple microscope, right? If you put a handle to it, it's what? A hand lens, right? A handheld lens. That is the simplest microscope we have. That is also a light microscope. Okay. Then, such as this one is a compound light microscope. What is the, let's say, the criteria to be a compound light microscope? At least 2 lenses must be there. At least 2 lenses must be there. Okay. What are the 2 lenses? Before that, let's learn about 2 parameters in a microscope. 2 parameters in a microscope, right? I'll write here. Yeah, those are the 2 parameters that we sort of how we evaluate microscopes, how powerful they are, right? That is magnification. අපි වාහනයක නේ horsepower එක, engine capacity එක, those things we say about a vehicle. Similar to that, to a microscope, we talk about its magnification and resolution power. Resolution power. Those are the 2 criteria. So the definitions of these you need to know, right? What are the definitions? Please highlight page 22. Magnification is the ratio of an object's image size. Online students, you can hear me, right? Image size to its actual size. Magnification is the ratio of an object's image size to its actual size. So if you write an equation, what is it going to be? Image divided by object. Yes, image size divided by object size. This is magnification. It's a ratio. It's a ratio. No. So always, what are we trying to do? Using a microscope, we are trying to generate a big image. Objects are very small. Cells එකක් වගේ, cell එකක් වගේ. It's a very small object. The image should be very high. Right? That's what we are trying to do. So when the image size increases, the magnification increases. So it's a good thing in a microscope. Okay. Highlight the definition of the magnification. Maximum magnification of a light microscope is 1000 times. Right? Usually, using a microscope, light microscope, we can achieve about 1000 times magnification. That means the image is 1000 times bigger than the object. That's the logic, no? Okay. Then resolution power is the minimum distance between 2 points that we distinguish as separate points. Highlight that. Resolution power. That means if you are sitting close to the board, you can see that these two lines are two separate lines. The lines separate lines there. But for someone sitting at the very back, they might see it like one single line. Because why? They are far away, right? That's because when objects get smaller, we don't see the details. Very details, right? So if you are not seeing this as two lines, that means you are missing some detail. No? But if you are seeing this as two lines, that means you are seeing detail. That is resolution power. Okay, this resolution power depends on what kind of wave we are using to observe, right? Light පාවිච්චි කරනවා නම්, in light, if you are using light as the wave, it limits the resolution power somewhat, right? Light use කරොත් the resolution power somewhat limited. One of the main reasons to adopt electrons for microscopes is the reason that is to increase the resolution power. Okay. So it says, page 22, resolution power of the light microscope is 2 micrometers. Right? After the light microscope again, minimum distance between two objects that we can see is 0.2 micrometers. These are MCQ points. Okay, highlight. It's a measure of the clarity of the image. Magnification is limited due to resolution, right? No matter how much you can enlarge the image, if that image is not clear, it's useless. Get it? So no matter how much you can magnify, if your resolution is low, then it's not useful. Okay. So keep reading. We are in page 23 now. Light from an object, the specimen, passes through the objective lens and produces a magnified image. Before that, for this this resolution power, you can say it is inversely proportional to the wavelength. This is lambda. Yes, this is used to indicate wavelength. No? So when wavelength increases, the resolution power decreases, right? So compared to your electrons, your light has higher wavelength. Therefore, the resolution power of a light microscope is low. That's why we adapted the use of electrons. Take this down. In your main tube, not in the practical tube. Okay, not in the practical tube. We have. I'll be giving a tube next week. Okay, right here we are here now. Microscopy. Okay, there's some space. Next page. There's some space here. So I'll give you some few things to write down. Write down the magnification equation and the resolution power equation here. Manage your space. Okay. Hold on. Anyone else who's new? You're new, right? Let me check. Ah, two tubes. Exactly, देखा. Magnification and the resolution power. Okay. Moving on. So they say in page 23, light from an object, the specimen passes first through the objective lens, then produces a magnified image. Then that image will be again magnified by a second lens that we call as the eyepiece lens, right? That's what's going to happen. So it's like this. So if we take this lens as your first lens, what is the first lens? That the light passes first through what? Objective lens. Light passes for the second time through another lens that we call as what? The eyepiece, right? So it is going to further magnify it like this. Yes. So here we have our eye, right? We are observer. Observer is here. This is what? Very small specimen. Yes. What is this? Draw this in the tube. This is the objective lens. Small line diagram, draw this. This is the eyepiece lens. Online students, your tubes will be delivered next month. Okay, once you have registered, will be delivered. I'll try to include this practical guide as well, printed copy of the practical guide and maybe some of the papers we did. Quickly draw this. Then we have a third equation that you need to know. This applies only for what? Compound microscopes. So with the combination of these two lenses, we can have a compound magnification. We can have a total magnification that is compounded, right? That has been increased. What is the equation of how you get the total magnification? Yes. How do you get it? Right? Total magnification is equal to, let's say, magnification of the objective lens and magnification of eyepiece lens. Okay. Write this down in the tutor as well, this equation. The total magnification of a compound microscope you can take by like this. So usually while you're taking down, this is the eyepiece, right? Eyepiece lens. Okay. So eyepiece lens, usually there's a standard. Eyepiece lenses are 10 times magnification, 10x, right? 10 times magnification. And in a standard light microscope, there are 3 objective lenses that you can rotate, right? Online students, can you see? This is an eyepiece. I'll explain the parts of the microscope properly, okay? Take this down quickly. So that's all that is there that you need to know from the resource book. So now we can move on to the light microscope practical. Okay, take out the tute now, practical tute. Okay, page number 3. Parts on functions of the light microscope and the use of the microscope to observe specimens. All light microscopes have 3 basic components. What are they? So now you know. Name some important components. What is the basic thing? First, you need to see the specimen, no? Right? You need to see the specimen. So what do you need? A light source. Right? First one, light down. Can you see 1, 2, 3? There are 3 numbers. You can say a source of light or a source of illumination. Right? First one, light source or source of illumination. Then what is the next important thing? Once you have given light, these 2 people, no, you need to have lenses, magnifying lenses. You have to have magnifying lenses. Third one, lenses උත් නිකම්ම තිබිලා හරියන්නේ නැහැ. Just having a lens, 2 lenses like this, it doesn't do anything. You can't see anything properly without what? Nicely focusing, right? Nicely focus කරන්න. So the third thing is means of focusing. Means of focusing. Means of focusing. You have to be able to move means of focusing. Okay, you have to be able to move the lenses ඉස්සරහට පස්සට, then to get a nice clear image, right? Means of focusing. What are the 3 things? Tell again. Source of illumination or light source, then magnifying lenses, then means of focus. Clear? Moving on. Okay, let's come to showing you the microscope itself, right? So don't laugh at my microscope. Okay, so basic thing, one of the most simplest things that you need to know about before touching a microscope is how you handle it, right? Right? You have to hold it like this, right? This part of the microscope, the curved part, is called the arm of the microscope, right? Arm. And this part where you keep the microscope somewhere is called the base of the microscope, right? You hold it from the arm and you support it from the base. Eyepiece it's going to break, right? Then you place it nicely on a flat surface. Okay, you place it on a flat surface where there is adequate illumination. Why do I need adequate illumination in this case? Because what is the light source? It's a concave mirror, right? Mirror එක, right? Then the basic parts. Okay, what are the two parts you know now? This is the base, this is the arm, right? Then where do we, so this is the light source, the mirror, right? The source of illumination. What is this? This is the place where we mount what we need to observe, where we mount a specimen. It's called the stage, right? This is the stage. Okay. And what are these? These are to hold the specimen in place like this, right? Give me a name. So if this is the stage now, these are stage clips, stage clips. Okay. Then we encounter our first lens. What is the first lens that a specimen is like the light waves are going through? Objectives, right? This is the objective lens. Online students, you can see, yes. This is the objective lens. Usually, in a compound microscope, there are supposed to be three objective lenses. But in this one, there's only one. Other two horugeni, right? It's literally, I don't know where they are. They are supposed two places where the other two lenses are supposed to fit. Okay, but in this case, there's only one objective lens, right? And this one, so this is objective lens. What am I? This rotating part, this rotating part that I'm rotating, it's called the nose piece, right? It's the nose, right? Because we can rotate it, it's called the rotatable nose piece. Okay, what is the function of the nose piece? You can change between different objective lenses based on what magnification you require. Okay, so usually there are three objective lenses. Usually they are I think 10x, 40 and 100. Those are the usual objective lenses, right? But in this case, there's only a 3-time magnification objective lens, right? Only a 3-time magnification. So I told you, this is the eyepiece, right? This is where you keep your eyes like this, right? This is the eyepiece, no? You can take them out. What did I tell? Its usual magnification is what? You can pass this around, right? Online students, this is the eyepiece, okay? Its usual magnification standard is about 10 times magnification. So in this microscope, what is the maximum magnification that we can achieve today? 10 and 3, 30. Using this equation, we can achieve a 30x magnification. Okay, right. So objective lens, nose, these are what? Your eyepiece lenses. They are, because this is a, let's say, bifocal. I mean, you can use your both eyes. We have two eyepiece lenses. Okay, clear? In a more complicated microscope, right? In a more complicated microscope like the one more complicated microscope, we have a like a electric light, right? Here instead of the mirror, we have to have an electric light. Okay, and also because we are having an electric light, we need a way of controlling that light. So there's a light, we can increase and decrease that light as well. So there's a switch, and also we can increase the size of the light beam that is coming through. For that, we have a diaphragm, right? There's a diaphragm. Okay, those are some additional things. I'll show you using the projector, a proper microscope. We are going to label it. Okay, so there's a diaphragm as well, right? Diaphragm is to increase the aperture, we say, how the size of the light beam that is hitting the specimen. Okay, then after the diaphragm, we have what? Yeah, yeah. Okay, basic things. Those are, and then we come to how we focus, right? A microscope ideally should have two ways of focusing, right? We have to be able to tattagala focus, right? Quickly we have to be able to get into a rough focusing, right? Lavata clear karaganna pataga. Then what do we have to do? we have to fine tune it, right? Nicely focus karganna one so that we can observe very clearly, right? So two kinds of focusing must be there. Two types of focusing, as rough focus and fine focus. Rough focusing and fine focusing killer, we have to have two types of focus. Okay, this is also called as coarse focusing, coarse adjustment or fine adjustment. We can also call. In this microscope, there's only coarse. There's only a coarse adjustment. Okay, there is no fine adjustment. You can only coarsely adjust it. Okay, that's another limitation. Right? Any other questions? So now you know the basic idea. Okay. So moving on, in the tube, eyepiece at the top is at the top of the microscopes and contains a lens with the ability to magnify about 10 times. Objective lenses are small cylinders located in the revolving nose piece. Usually 10 times, 40 times, and 100 times magnification objective lenses are there. Okay. Total magnifying power is taken with this equation, objective into eyepiece. Then resolution power and magnification are important parameters. You can write down the definitions here. Okay. Can you see this space? Magnification is, please write down. What's the definition of magnification? And write down the definition of resolution power as well. Those are definitions that you need to know by heart. Okay, very commonly questioned in the structured essay paper, right? Definitions are by hearting, right? There's no other option than to by heart, because if you, I have told you this again and again, if you miss a keyword, no marks. This is a rich microscope, right? Not like mine. I mean, it's a complete one. Online students, hold on. Okay. So using this, let's do some proper labeling, and then you can draw one of your own, right? So you have the same diagram in your tubes, right? So previous page. So labeling should be done there, right? You can label this diagram. What are the things you can see? Yes, what are the things you can see? Things you know now. This is what? The base, right? The base. Okay. Right. Then the base using supporting the base and holding from the where? This is the, yes, label with me, arm, base and arm. This is what? Stage. What are these things that are used to like fix the specimen on the stage? Stage clips. This is what? This is a electron, I mean, sorry, it has a like a electric light source. So this is, you can say, light source. And there are some, there are two additional things that I sort of I want you to know. This is sort of the bottom, you have a bulb, right? Here you have the bulb. And there are two other sort of gadgets that are helping to sort of use that light nicely. There's what we call as a diaphragm. Diaphragm, it's like this. Have you seen in cameras, there's an aperture, no? Right? It's an opening like this that you can sort of, this can be made bigger and smaller. Like photos, it opens and closes, right? It's like this. The diaphragm you can control. So you can like control how big the circle of light coming from the bulb is, right? You can make it very big, or we can make it very pointy, sharp, using the diaphragm. And similarly, can you see here? There's another lens. Don't be confused. Okay, this is not used to magnify. This is called the condenser. That is used to again to like, that is used to focus that light into the specimen. Okay, that is the condenser and the diaphragm that is missing from my microscope. Okay, label those. So stage clips. What are these? You can see the three properly, right? Online students, you can see, right? What are these? Objective lenses. Yes, these are objective lenses. Three. The biggest one is likely to be 100. No. Then this is the shortest one. This is the medium one. Can you see? So this is maybe like 40. Sorry, this is maybe like 10. This is maybe like 40. Right? What are they attached to? Rotating nose piece. Online students, you can see, right? Rotating nose piece. This is what? Eyepiece. Then two types of focusing. This one has the them properly, right? This is your, the big knob is your what? The big knob is your close focus. Close focusing knob is the proper answer. This one, okay, big one, close focus, and this small one is what? Yes, fine focusing knob. Okay, quickly take this down. Label the microscope nicely. Right. So homework, you have to draw a microscope by yourself. Okay, in this space, draw a microscope by yourself. You can find a nice line diagram of a microscope. This is what I've been studying. Okay, pharmacology. So reminding me to study. Right. Right. So in your practical guide, did you download the practical guide? Right, from the NIE website, you have the practical guide PDF. Okay, A-level practical guide. It's better if you can, I mean, just have that PDF with you. Okay, so in there you have a simple line diagram of a microscope, light microscope. Take a line diagram method. So if you can't draw one by yourself nicely with all the parts, you can copy it. Right. Homework, online students also, in this space, draw a microscope. Okay. First term test maybe will be question to draw a microscope, not for the A-level. Okay. So better practice. Okay, page number 4 of the tute. Next page, there's a chart that we are going to fill now. You need to know what are the functions. This first thing, that is the main switch, that is not that necessary, right? Used to switch on off the light source. Just write it down. Used to switch on or off the light source for main switch. Light intensity knob is again a bit unnecessary, but you can say adjust the light intensity. Have you seen, you can make it bright and low, no? Right? Those kind of filament bulbs that you can control. Adjust the light intensity. Then what's next part? Eyepiece. What do you know about the eyepiece? Is he magnifying first or second? Imagine the light is coming from what direction? Down to your eye. So who is the lens that is first magnifying? Objective. Eyepiece, write down, magnifies image for the second time. Magnifies image for the second time. Field of view not necessary. Keep it blank, right? It's what you can see, right? Then condenser. Condenser. What do you mean? What is the function of the condenser? It's again a lens, right? You can say focuses light from the light source. Focuses light from the light source. Focuses light from the light source to the specimen, onto the specimen. Onto the specimen. Then cut the diaphragm knob, cut the knob part of it, just the diaphragm. Cut the knob. The diaphragm controls the amount of light reaching the specimen. Diaphragm controls the amount of light reaching the specimen. Online students, you can hear, right? Yes. Any questions you can send now. Then stage. Used to mount the specimen. Used to mount the specimen. Used to mount the specimen for observing. Mount the specimen for observing. Improve your writing speed a little bit as well, right? It will be important. I mean, for in bio, if you don't know, then you won't be needing to. I mean, if you don't know the essays, then you don't need to write very fast, right? But if you know, but you can't write very fast, that is unlucky, no? So for some luck, you got all the 4 essays from things that you studied very well. So you know what you need to write. But if you run out of time, then so improve your writing speed a little bit as well, but keep the clarity, okay? Don't lose the clarity. Lassana onne legible, right? That's what's simple. Right, where are we? Stage. cut specimen holder and write stage clips. Cut specimen holder and write stage clips. Okay. What is their function? Hold the specimen on the stage. Hold the specimen on the stage. Main switch is to switch on and off the light source. Okay, don't think about that much. Okay, specimen holder, cut it and stage clips. Okay, revolving, or better say rotating. It's not revolving, right? Revolving means like rotate, right? Rotating nose piece. Stage clips to hold the specimen on the stage. Stage clips. Yes. Yeah. Right. Hold the specimen on the stage. Then revolving nose piece. What is the function? To change the objective lenses. Change the objective lenses to the field of study. Change the objective lenses to the field of study. So when you need to use the low power, you rotate. Then medium, high, you just rotate the nose piece. Then you can change the objective. Next, objectives for 1000. It has to be a 40-1000. Okay, 40-1000. Magnifies image for the first time. Magnifies image for the first time. Magnifies image for the first time. Close focus adjustment knob for rough focusing. For rough focusing of the image, right? For rough focusing of the image. By making large changes. Large changes. By making large changes. Can you see? So when you move it by a visible amount, it moves, right? The eyepiece and the objective, everything, it moves by a visible amount. So if you, in a fine focusing, actually to use the fine focusing, we have to lock the coarse focus, right? We have to, there's a sort of in the coarse focus, there's a locking mechanism. You have to lock it, otherwise you can't rotate the fine focus, right? Have I told you this in our main first 2 years again, right? Because those are the interesting subjects that we had, right? So for anatomy, subject of anatomy, we study histology under that, right? For anatomy, we have what we call as a spot test, right? Spot test very stressful. Okay, here tables in the dissection hall where we dissect the bodies, no? Right? Exam we are given a piece of paper. Numbers then there are specimens on the table. So, right? Usually 10 specimens. Right? It can be a head, it can be a leg, it can be 2 microscopes, then it can be X-rays, CT scanners, CT scans, and what else? Yeah, several body parts, right? An abdomen, chest, heart, or whatever. Slices can be there, whatever can be there. And there are labels put into them. You need to identify, right? So one station, it's 30 seconds. Okay. And there are definitely 2 stations with microscopes. You need to observe and identify what is there, and you have to write them, right? So in the exam, actually, the microscopes are locked. The coarse mechanism is locked. Coarse focusing is locked, right? අපිට ඒක අල්ලන්නවත් දෙන්නේ නැහැ. right? In the exam, we are not allowed to touch the coarse focusing. We are only allowed to touch the fine focus. Because why? The ඔයා ගිහිල්ලා මේකේ coarse focus එක change කරොත් ඊළඟ මනුස්සයා in the 30 seconds, maybe 20 seconds will be he has to sort of change the focusing, right? So the rough focusing will be done for you, but depending on your vision, right? කොච්චර පොට්ටද කියන එක අනුව you have to sort of adjust somewhat. No? So that will be done by the fine focus, right? You're only allowed to touch the fine focusing. Rough focusing අල්ලන දැක්කොත් කියනවා ඊළඟට යන්න, then the exam will be paused and until the focus is correct, right? And it's stressful, like ටංගාලා බෙල්ලයක් ගහනකොට we have to jump to the next station. Just ටංගාලා ගහනවා, we have to jump to the next station, right? It's very stressful. And එකක් කෙලවුණොත් දිගටම ඉතින් අර අන්තිමට first question එක හරි second එක හරි if you didn't know ඉස්සම අවුල් ගිහලා අන්තිමට එළියට යනවා හිස් කොලයක් අරන්, right? So as I was saying, coarse focus, it will lock. Then to use the fine focus, what is the purpose of the fine focus? Right? Focus the image, write down, focus the image by making small. Okay. So those are the paths and the functions you need to know. Online students, any questions? No? Then go home and read the proper handling of the microscope, right? The next things in the, these are something additional for you, right? So you don't need to know the process, okay? Go home and read that. Right. Then last page, we have a blank page, right? So let's write down the process of the practical that we are going to do today. That is, in your syllabus, it is either observation of onion epidermis or rohio. So no one, I didn't have rohio at home, right? So I brought an onion, and already there's an onion prepared one. We can observe that, right? But before that, there's a series of steps that is also questioned in the tute, right? Take the main tute, the main one. In the main tute, under the questions that we have about microscopes, oh, no, it's not there. It's not there. Sorry, right? Anyway, it can be questioned in the structure. They say the steps, the series of steps that you need to follow to do this practical properly. There is the observation of onion epidermis. Okay, let's write down the steps. Let's write down. Yeah, any question? No? Number one, first step, write down. Online students, write down. Make thin epidermal peels. Make thin epidermal peels of onion or rohio. Of onion or rohio. It's scientific. I mean, rohio is like this. Onion slash rohio. And place it in water. Make thin epidermal peels of onion or rohio and place it in water, preferably, no need to say prefer, within brackets, in a watch glass. In a watch glass. In a watch glass. Online students, also write down. Transfer the onion piece. Transfer the onion piece into a drop of water into a drop of water on the center of a drop of water on the center of a clean glass slide, clean glass slide, clean glass slide using a paintbrush. Transfer the piece of onion into a onion peel, okay, into a drop of water on the center of a clean glass slide using a paintbrush. Okay. Hold, next one, hold the cover slip, hold the cover slip at the edge of the drop of water, hold the cover slip at the edge of the drop of water with the help of a mounting needle, with the help of a mounting needle, comma, and gently lower the cover slip, gently lower the cover slip while supporting it with the needle, while supporting it with the needle onto the drop of water. Okay. Onto the drop of water. Do not let air bubbles be trapped. Do not let air bubbles be trapped under the cover slip. Cover slip. Under the cover slip. Place the slide. Next point. Place the slide on the stage of the microscope. Place the slide on the stage of the microscope. And observe under low power objective lens. There's a disturbance. What do you mean? Can you hear me? Okay, sorry. No, I'll repeat. Place the slide, no, looking through the eyepiece, smooth the slide to bring into position of study. Okay. Okay, all right. Looking through the eyepiece, move the slide to bring it to the position of study. Then Which one do you want me to repeat? Okay, keep space. I'll go through them again. Okay, keep writing. Next point. Adjust the mirror. Adjust the mirror. Adjust the mirror. Condenser, diaphragm, condenser, diaphragm to give optimum illumination. To give optimum illumination for the object. Optimum illumination for the object. Use the coarse focusing knob. Use the coarse focusing knob to get the image clear as possible. Use the coarse focusing knob to get the image clear as possible. Then use the fine focusing knob. Next point, use the fine focusing knob to improve the quality of the image. To improve the quality of the image. Next point, using the rotating nose piece. Using the rotating nose piece, bring medium power or high power objectives. Bring medium power or high power objectives to increase magnification. To increase magnification further. That is the practical. Okay. Right. So what we can do next is you can take turns and come and observe. Let's try to prepare onion peel. Anyone who's willing to donate their mouth lining, buccal lining, that's part of the practical. Cheek epithelial cells inside. If you can, we can. There are no yogurt spoons there either, right? So using a yogurt spoon, we can scrape and put it on a drop of water. Then we can observe, right? Okay, right. For now, let's try to observe this, right? And the thing is, this one doesn't work. Okay, right object, the eyepiece doesn't work. There's a prism inside that is broken. Only the left one is usable. So using one eye, I'll adjust the light source for you, and can't see the specimen. Okay, light is not enough. We might have to use a torch. Right? Right. So it's very primitive, but don't mind. been a bit dried out, so the cells are dehydrated, right? Come one by one, let's try to prepare a new slide as well. And Colin. Okay. Let's try to. So cells are a bit deep, very sort of tightly packed. Okay. Shooty loonu gediya tibba. So in our when we did this practical also, no one brought onion, so we had to buy like a burger and get the onion peel in that, right, from the canteen, right? Right. මෙතනින් චුට්ටි පීල් එකක් ගන්න ඕනේ. Anyone who has the skill? අත කපා ගන්න නැතුව පුළුවන්. පීල් එකක් අරන් දෙන්න. Anyone who can nicely peel me a slice? මේකට තමයි දාන්න ඕනේ. I'll put a drop of water in there. මම කරන්න දෙනවා. I think this one is nice, huh? So Emma, I don't have a watch glass. You don't need exactly a watch glass. So you transfer it. Can you see? You transfer it to the glass slide and put another drop of water. Yeah, any question? No, right? Some drops of water. Online students, you can see, right? So have you seen cover slips? Right. So you take a cover slip and then there's something called this needle that is used to, what is the name of the needle? What did I give? What needle? Mounting needle. Right. You're supposed to use a mounting needle to make sure that there are no air bubbles. Okay. But no need that, right? edge එකේ ලස්සනට තියලා. So slowly if you can put it down and it's kind of okay. Right? I think it came out okay. Let's see. Ah, yes, this is better. And ladies first. This specimen is better. Come. 12:30 ඉවර කරන්න. Yes? No. About the early practicals, online students? Yeah. I mean, not that clear. Okay. Not that clear because why? Our magnification is limited to only 30. Right? Only 30x magnification is possible. Using a hand lens also you can achieve about 10 times. Right? Using a hand lens you can achieve about 10 times. Okay. This is just 30 times. Just to show you the structure. That's all I want.